Okay, so hi everyone. Finally a physical conference, and therefore experimental tribute to this great meeting. This is an experimental measurement in a microscope, essentially with the conference name. But before we get into all of that, let me first introduce myself. So my name is Elias Nehme, and I'm from the Electrical Engineering Department at the Technion, jointly supervised by Professor Michaeli and Professor Yoav Shechtman. And today I'll be presenting our work titled Learning Optimal Wavefront Shaping for Multi-Channel Imaging. Okay, so first let us start with the motivation. So essentially in biology, several interesting applications require us to track the subcellular diffusion of subcellular elements. And now this information can tell us a lot about cellular function. Specifically in sleep and cancer research, we're interested in tracking telomeres, which are the tips of chromosomes inside the nucleus of our cells. Now, naturally, biology is three-dimensional because this is the way things are built. However, in a standard microscope, we acquire a 2D image only. Now, usually to see these telomeres, we tag them with fluorescent proteins, and therefore a time-lapse image of such a tracking behavior would look something like this. Now, as you can see, only a few points will be in focus in a standard microscope, and the rest will be defocused simply because they're at a different depth than the camera focal plane. And this is a problem because these things are moving, so we can't simply just scan the focal position to get their to get the entire 3D volume. And therefore, the question is, how can we image and track in 3D, and so getting 3D tracks over time? So to answer this question, let us look more closely at a single point source moving out of focus in a standard microscope. Okay, so here you see essentially a point source moving in Z at the left, and here you see the PSF, or the image of this point source, being changed while this point source is moving. And as you can see, the image of this point source, or the PSF on the camera, is symmetric above and below focus, and therefore we have a depth ambiguity. We can't tell if it's above or below focus. And also what you can see is that we have a very narrow Z range, that after this Z range, the signal becomes very diffuse. And therefore, in typical SNRs, this will be drowned in noise. So the trick to remedy this is to manipulate the Fourier plane, which is here, by inserting an optical element called the phase mask, such that now, after inserting this phase mask into the system, the shape generated on the camera for a single point source is not symmetric above and below focus, and therefore we don't have depth ambiguity. And also, the signal is preserved very nicely across the entire Z range. Now, specifically, this example PSF is called the tetrapod PSF, and was shown to be optimal for estimating the position of a single emitter over large Z ranges. Okay, so now coming back to the 3D tracking application we had from earlier, applying this trick in the Fourier plane, we get time-lapse of images that look something like this. Now, you see that all of our out-of-focus emitters have a distinct shape telling their depth, and therefore we're truly acquiring with a single snapshot the entire 3D volume. However, if we look at nearby points, we see that their shapes overlap on the camera, and therefore this makes it difficult to precisely tell their 3D positions. So in our work from Nature Methods last year, we essentially learned the PSF end-to-end with the reconstruction network to try and overcome this challenge. And indeed, we were able to push the detection performance at high emitter densities from this tetrapod baseline significantly higher up. However, despite the great advancement, there are still lots of very important applications that live somewhere around here. And therefore, the question of this work, the premise of this work was, how can we get higher detection rates at high emitter densities? Okay, so the main proposition in this work is the following. Instead of capturing 100% of the signal in a single imaging channel, getting a fairly high SNR image in camera 1 here, what we do is we do the following. We give up on this SNR slightly and get only 50% of the signal into this imaging channel, and then tunnel the other 50% into a second imaging channel employing a different PSF. And therefore now we have two images, although in each channel we have strictly reduced SNR, but we have twice the sensor area. And therefore, if we design the PSFs cleverly, maybe we can increase the detection rate at high emitter densities. Before I tell you how we design the PSF of each channel, let me first tell you how, given a pair of images, we extract the 3D positions. And this is done by a convolutional neural network trained on simulations. Now, specifically, this architecture is not a standard one. We proposed it in our work from last year. But what you really need to know about it is that it takes the concatenated 2D images and output a 3D occupancy grid where occupied voxels are essentially emitters, stands for localized emitters. Now, to train this architecture, we use a loss function that is comprised of two terms. Okay, the first term, it tries to measure accurate localization, and the second term, it tries to increase the detection rate. Okay, now that we know how to localize the images, the true question is, how do we acquire the two images in the first place such that we get high detection rates at high emitter densities? Now, to answer this question, we propose three different design methods. And first, let us start with the intuitive one. Okay, so in the first method, we design an extended depth of field PSF, or an EDOF PSF, that maintains a Gaussian-like spot across the entire Z range, regardless of the emitter depth. And we combine this PSF with the tetrapod PSF. So intuitively, we're splitting the localization problem into complementary channels, where the EDOF PSF encodes XY and the tetrapod PSF encodes Z. And so now if we look back at the overlapping tetrapods in dense regions, and we're assisted with this additional measurement with the EDOF PSF, then this measurement essentially can act as an oracle, giving away the XY positions of all the emitters in the overlapping region. And therefore, the network can use this additional information as prior knowledge to solve for Z in the overlapping regions in camera 2. So that was, in a nutshell, method 1. Moving on to method 2, we take an estimation theory point of view. Specifically, we minimize the Kramer-Rao lower bound for a single emitter position estimation. So here you see the phase mask being updated in the Fourier plane and the PSFs being updated in the image plane. And as you can see, this is the stable local minima of this optimization. Now, if we look at this result more closely by again scanning a point source in Z, we see that essentially the information is split axially between the two channels, where one channel encodes for positive defocus and the second channel encodes for negative defocus. And this is also evident in the Kramer-Rao lower bound plots, where one mask optimizes the lower part of the axial range, the second mask optimizes the upper part, and jointly, they both optimize the CRB efficiently over the entire Z range. Finally, for the cherry on top, we design the PSFs with the most successful method, which is the end-to-end learning. In this case, we simulate 3D positions of overlapping dense emitters and pass them through the numerical model of our microscope to generate the intensity patterns. Afterwards, this is split by a polarizing beam splitter such that it's fed into two encoders, which are essentially physical simulation layers, each parameterized by its own phase mask, calligraphic M1 and calligraphic M2, and the resulting encoded measurements are then concatenated and fed to a reconstruction network. Now, this network tries to recover our simulated ground truth positions, manifested here by these green triangles. And using our loss function, we compare the recovery of the network with the simulated ground truth and backpropagate the gradients, both to update the reconstruction network and further backpropagate through the physics to update the phase masks at the Fourier plane. And we repeat this essentially until convergence. So here you see, again, the phase mask being updated in the Fourier plane and the PSFs updated in the image plane. And as you can see, very quickly, the network decides to converge to complementary patterns, although our loss function is, or although our optimization is performed pixel-wise, and we don't have a loss function enforcing this behavior. Okay, and this persists throughout the entirety of the iterations, such that essentially the resulting phase masks in the Fourier plane are almost flipped versions of one another, resulting in complementary PSFs in image space. Now, if we look at this result again more closely by scanning a point source in the Z direction, we essentially see that this is the resulting PSFs. So we see a very interesting behavior here, such that the 3D position information is multiplexed into the two channels. And if we look at the composite of these two channels, we see that essentially the side lobe movement between the two channels are cooperating such that the depth is encoded in the relative movement. Okay, so now that I have bombarded you with 3 different methods to design the PSFs, the true question is, how do our results compare with the current state of the art? So to answer this, we measure detection and precision in realistic simulations. And specifically here you see again the current state of the art with the end-to-end optimized single channel PSF. And this is the result of our 3 methods, all improving in detection compared to the current state of the art. Specifically, for method 3, we get almost a 2-fold improvement at the highest tested emitter density, which is a great result. As for precision, and this is the current state of the art, and in here actually we measure precision by RMSE laterally and axially. And in this case, you would like your RMSE to be lower, because this means more accurate. And this is the result for our 3 methods, and as you can see, method 2 and method 3 improve the results, where again, method 3 is the leading approach. Now, just to give you a hint what this density axis really means, let's take a visual example at a density of 0.5. So this is simulated with method 3 with the PSFs learned end-to-end. And if we feed these two images into the reconstruction network, we get a perfect reconstruction of all 90 emitters with a Jacquard of 100%. And the lateral and axial RMC are also very low, which means very precisely. Okay, so showing you lots of simulation is nice, but the true question is, does this work in real experiments? This is where the catch usually lies. Okay, so to answer this question, we performed a live cancer cell experiment where we track the telomeres of a cancer cell over time. Okay, so here you see the captured images in the microscope, and here you see the estimated nuclear envelope, the localized telomeres, and the tracking time bar. So if I play this movie, you can see that essentially the network is able to localize all of the visible telomeres very accurately, tracking all the different diffusion behaviors across the entirety of the nucleus. So to validate that, in fact, our 3D positions are not just made up 3D tracks, we came back to the experimental data and rendered our 3D positions recovered by the network using the imaging model. Okay, so here you see the two imaging channels, the experimental measurements. On the top right of each, you see the measured zoom in the white square. And below it, you see the recovered image by the network. And if I now scan this white square across the entire field of view, you see that the PSFs are very much matching in both channels, which means that we're, in fact, precisely recovering all positions. Okay, so after such a long slide, let us just conclude. So to summarize, in this work, we proposed a dual view scheme that splits the intensity 50-50 between two cameras in order to increase the emitter detection at high densities. And specifically to design the PSFs of both cameras, we proposed 3 different design methods, where the leading method was the end-to-end learning of both PSFs. And now coming back to the original premise of increasing the detection rates at high emitter densities, with this work we were able to push this significantly higher up. And therefore, if you're a biologist interested in such things, there are exciting new opportunities for you using our method. And with that, I would conclude and thank all my collaborators and, of course, the funding agencies who made this work possible, also Daniel Freedman from Google. And if you're interested in more details, please refer to the paper. Also, code and sample data will be soon on my GitHub, so stay tuned for that. And finally, I will thank you all in an experimental fashion yet again. And with that, if time permits, I would like to take any questions.